Hello folks, this is Required Practical 9, which is an investigation of the effect of temperature on respiration in yeast. So here's some yeast, this is what we're going to be looking at, and we've mixed this yeast with a solution of glucose, so it's got some respiratory substrate, and we are going to be testing different temperatures to find out how it's respiring. We're going to measure this using a chemical called methylene blue, which is unsurprisingly blue at the moment. This is it in its pure form. We've diluted it slightly to make this indicator in here. Now methylene blue is normally this colour, but when it gets reduced it goes colourless. It's a bit like the DCPIP that we use in one of the photosynthesis experiments, because in both of those reactions things are getting reduced. In respiration you should be aware that NAD is getting reduced to NADH, and if this chemical is hanging around then it might interfere in that process, get reduced itself in place of the NADH, or the NADH itself might reduce the methylene blue. But either way, it is going to go colourless, and the more respiration's happening, the quicker that's going to go colourless. So it's a really useful tool to measure the rate at which respiration is taking place. So in this experiment, you're going to be using yeast at different temperatures, and you've got methylene blue. So think about your risk assessment. Obviously, eye protection as always. And yeast is potentially an allergen. It's a living organism. If it comes in contact with your skin, you don't know what's going to happen. So allergen is something you need to consider. If you get it on your skin, of course, you will wash it off. And this is potentially an irritant as well. So bear that in mind when you think about risk assessment. Now, I'm going to demonstrate one of these, and I'm going to start at one temperature. And I'm going to leave you to think about the other temperatures you might choose. So I'm going to do the first experiment, just to demonstrate it, at 35 degrees. And the first thing that you need to do is you need to get 3 test tubes, and we are going to put 2 centimetres cubed of the yeast and glucose mixture in each. So here we go. Here's my syringe that I'll be using for this, and 2 centimetres cubed is going into each one. There we go. You might have noticed that I gave that a bit of a stir before I drew that up. You might want to think about why that is. This is a yeast suspension. There are lots of cells in it, and I want to try and get a nice even distribution of those yeast cells so that each of these tubes is going to have the same number of respiring organisms, hence the stirring. Right, so there's my 2 centimetres cubed of that into each one. And it suggests in the method that you label these. Now think about where you're going to label them. They will be going in a water bath, so if you label down here, you run the risk of the label floating off. So here we go, I'm going to label them 1, 2 and 3. Okay. And now what we need to do is we need to get those respiring organisms up to the right temperature. So I'm going to be using a kettle for this, but given that my starting temperature I'm going to be using is 35 degrees, I probably don't need to boil it all of the way. So what I'm going to do instead is I'm just going to partially boil that, and I did that a moment ago. It's now sat there kind of lukewarm, and I'm going to use a combination of that and some cold water to get my water bath at the right temperature. So there we go, there's my warm water. Cold water, okay, and I'm aiming for 35 degrees. So this is just a water bath, and I want it to be a nice consistent temperature. So just leave that for a moment to just check it's equilibrating. A little bit on the high side, so I'm going to add a little bit more cold in. And hopefully can see that we are pretty much just a smidge above, but we're pretty close to 35. Let's give that a moment to cool down again. Okay, tiny bit more cold. That's pretty good. So now what I've got to do is I've got to put these guys in, and I've got to wait until the contents are at 35 degrees. Now this is going to take a few moments, and obviously this is just sat on the desk at room temperature. It's going to cool down. So you're going to need to do a bit of juggling, adding small amounts of this, stirring, and of course you shouldn't stir with a thermometer like I just did. You should use a glass rod, because it's less expensive if you break it. Well, that's going to take a few moments. I need to make sure that my yeast is at that temperature, my starting temperature, because that's really important. That's the variable we're changing. So I'm just going to leave that for a second. Right, I am now reasonably confident that my tubes and their contents are at the target temperature of 35. So now my next thing is I'm going to take 2 centimetres cubed of methylene blue. Okay, I'm going to add it just to test tube 1. Right, what you can see is, give it a bit of a mix, that it goes this lovely kind of slightly off greeny blue colour. I'm going to pop that back in there. I'm going to keep an eye on it because remember, decolorization is what I'm looking for. That is going to take a bit of time to decolorize. I've started a stop clock and we are going to see how long it takes to return to the original colour. Once I think it's decolorized, I can stop this and record that as my first data point. Okay then, so the experiment's been running for a few minutes now, and if I just show you what's happened to the colour, you can hopefully see that that blue colour that was completely throughout the entire tube has now kind of largely disappeared from it. There's a little bit of blue at the top, and we'll talk about that in a moment. So I'm going to judge that as having reached the end point. So we've had decolorization, so I'm going to stop the clock, and I've got a time of 2 minutes 35 seconds. So let's write that down. Okay, so I've recorded my first result. Now I've also got tubes 2 and 3. Now they are still at the required temperature. So what I can do with them is I can now set up another repeat. So 2 centimetres cubed, methylene blue. In it goes. Of course I'm keeping my eye on the water bath, making sure that the temperature is okay. So this one I'm now ready to start, and that'll be my repeat. Of course, if you were feeling time efficient, what you might want to do is you might want to start 2 simultaneously. Although technically I started those 10 seconds apart, but you keep an eye on those and see when they decolorize. Now, things to consider when you're doing this experiment. This is the one that's finished here. And as you can see, it's not completely decolorized. There's a bit of blue at the top, and there's the reason for that. Remember, methylene blue, when it's reduced, it goes colourless, and the opposite of reduction is oxidation. So if it gets reoxidized, it will go blue again. And the top of the tube is coincidentally where oxygen is available. So you're never going to get that bit of colour disappearing completely. One thing you must never do with this experiment is, whilst it's sitting in the water bath, you mustn't pick it up and give it a good shake. You might do that in the hope that that kind of speeds up the reaction. But all you're doing there is you're stirring the oxygenated layer back through. You're actually giving oxygen to the rest of the methylene blue in there. And you might actually turn it blue again. So you should leave it still for the whole thing. The really tricky part with this experiment is deciding when it reaches the end point. Now this one's turned a little bit blue again. Remember the yeast that we started with looked a little bit like this. And if I dilute it with something that's completely clear, so there's no blue colour here at all, just a bit of water. Okay. This, I guess, could serve as a reference tube. You'll be able to use this to determine when you think the blue tube has reached the end point, but it's difficult, it is subjective, and you'll need to consider that in your evaluation. Using this setup, you'll be able to get 3 sets of results for 35 degrees, and then you'll need to choose some other temperatures to test. So I'm just going to pause the tape there, collect some more results, and then we'll discuss the ranges that we might use. Okay folks, so here are my results for that one experiment. 3 repeats. You would be able to calculate a mean average from those. But what of course you need to consider is the temperature range. We started at 35, and you need to consider if that's the temperature we started at, where do we go from and to? Can we start much lower than that? Will we start much lower than that? And what do we go up to? What would a sensible temperature be to reach? Now, you need to think about this quite carefully, because obviously, theoretically in the lab, we could do everything between a temperature of 0 and using a kettle, you might just about get up to 100 degrees centigrade. You need to consider which bit of that range you want to investigate, which is the sensible bit. You are looking at the action of those enzymes in respiration, so consider a sensible range, and also consider a sensible interval to go up in. Okay, so hopefully you've had a little think about the range you might want to use. I've just got some tubes here that are still decolorising. They're at a slightly higher temperature now, so we're up to I think about 50 degrees. Of course, what you might want to consider whilst you're doing this experiment is that you need to have a control experiment running at the same time. Now remember, a control experiment is set up so that you can just confirm that any changes you observe are due to the active biological factor, in this case the yeast cells. You want to make sure that this methylene blue doesn't naturally decolorise on its own. So in order to do that, you are going to need to give it some yeast that is not going to be respiring, that is not active anymore. So this test tube here, this has been out on the side now for well over half an hour, and it's actually got some boiled yeast in it. So at the very start of the experiment, I popped this in a water bath with boiling water in, and I left it there for quite a few minutes to ensure that that yeast was completely boiled. The enzymes were completely denatured, and as you can see, it still has this green colour. It looks the same as those test tubes I've just started off on the experiment, and yet this has been there for 30 minutes. If I leave those for 30 minutes, I fully expect those to decolorise, but my control experiment does not. So you need to factor that into your plan as well. Make sure you have a suitable control. Okay, that is required practical 9. I hope that was helpful.